Dr. Tshetiz Dahal. I am from Lugansk State Medical University, Ukraine. And I'm very thankful to Alzheimer Disease Live Conference. And my title today is for Lapse Memory Deficiency in an Alzheimer Disease Model using Physical and Cognitive Exercise.
Yeah. So,
can we move to next slide?
Sure. Please. The stage is all yours.
Okay, sure. Thank you. This is my details of you. Let's move to abstract. Alzheimer disorder actually is a leading cause of dementia within the global, accounting for 50 to 75% of instances. Currently, there may be constant treatment for Alzheimer diseases, but the present day pharmacological therapy minimizes symptom development but does no longer reverse brain damage. This research is mainly focused on non-pharmacological treatment for Alzheimer disease that was to advance to behave on brain plasticity and minimize the neurotoxicity caused by the amyloid beta peptide. The use of neurotoxicity version precipitated through AB in rats. The present look at shows that bodily, physical exercise and cognitive workout reverse recognition reminiscence deficits with outstanding impact of long-term object recognition memory. Decrease hippocampal lipid perioxidation, repair the acetylcholinesterase activity altered by using AB neurotoxicity and appears to reverse as a minimum in part hippocampal tissue disorganization. The keywords are Alzheimer disease, physical exercise, cognitive exercise, oxidative damage, AB neurotoxicity. These are the brain scans which help identify Alzheimer disease. The left side is for normal and right side is for Alzheimer's. It's done with positron emission tomography, PET, also known as. It affects brain activity, how, like you can see in this slide, the normal one and the Alzheimer's one. The blue and black areas in the right image indicate reduced brain activity resulting from the diseases. According to WHO, AD is a global health priority. This neurodegenerative disease is the most common form of dementia, accounting for 50 to 75% of cases. AD is related to aging, and every 5 years, after 65 years of age, its prevalence doubles. The formation and aggregation of normal amyloid beta AB peptide in the extracellular space, hypophosphorylated TAU protein, and brain oxidative stress are some of the pathological alterations found in AD. These alterations lead to a gradual loss of cognitive function, usually starting with short-term memory dysfunction, impaired judgment and reasoning, the disorientation, and culminating in total memory loss and personality alterations. In addition to high prevalence of AD, there are limited options for treatment of the disease. Furthermore, the current pharmacological therapies only minimize the progression of the symptoms. They do not reverse brain damage. In this sense, research focused on non-pharmacological treatment has been developed. Among the possible non-pharmacological strategies in physical exercise improves cerebral blood circulation, thereby increasing the supply of oxygen and energetic substrates to the brain. The effects of aerobic exercises, such as running on a treadmill, can also be related to its effect on reducing the formation of amyloid plaques and the hypophosphorylation of TAU, and on reducing neuroinflammation and oxidative stress. Another potential non-pharmacological treatment is cognitive exercise. Cognitive training contributes to maintaining neural functions, promoting cognitive flexibility, decreasing oxidative stress, and improving the quality of life in patients. Evidence suggests that CE produces long-lasting improvements in the memory performance of older adults who experience a normal cognitive decline. Previous studies have demonstrated that cognitive stimulation programs are effective in maintaining cognition and quality of life in AD patients. with mild to moderate dementia. Despite these positive observations, the mechanisms by which CE acts in AD brain are not yet well described. Recently, using an animal model of AB neurotoxicity, we showed that CE is effective in protecting against memory deficits when it is performed before neurotoxicity induction. In this case, we observed that CE was able to avoid the brain oxidative imbalance induced by AB. One of the main properties of the brain is neuroplasticity. The term neuroplasticity refers to ability of the organ to change according to external stimuli, altering its functions and morphology through neural mechanisms such as synaptogenesis and neurogenesis. Learning and memory are examples of functions that are highly dependent on hippocampal neuroplasticity deficits in this process, as occur in AD, lead to memory impairments. Cognitive plasticity may be strengthened via aerobic and cognitive training. It is important to highlight that our group has already demonstrated that CE is a good SPE as a preventive strategy for memory deficits related to AB neurotoxicity. Despite the well-demonstrated preventive effects of PE and CE, we investigated whether the same strategies could be used to treat memory deficits. In the present case, PE and CE training were introduced after the introduction of AB neurotoxicity when cognitive deficits were already established. Our results demonstrate that PE and CE can reverse memory deficits, hippocampal oxidative imbalance, and some hippocampal morphological alterations related to AB neurotoxicity. So the methods and materials are goes like animals and experimental design. Adult male Vista Rats were purchased from the veterinary hospital, Government of Sikkim, India, and were housed at Research Center, vivarium under controlled temperature, 23 plus 2 degrees Celsius, in a 12-hour light dark cycle with food and water available. Initially, the animals were divided into two large groups and were subjected to stereotaxic surgery for intrahippocampal infusion of AB protein, a saline vehicle, followed by 10 days of recovery from surgery and AB aggregation. After this period, animals were subdivided according to the treatments, PE training, CE training, or no treatment, resulting in 6 groups. N by 12 group, sham surgery control, AB-induced neurotoxicity model, sham surgery and PE training, sham surgery and CE training, AB and PE training, and AB and CE training. After that, the animals were subjected to behavioral tests to evaluate recognition memory and to monitor control parameters. Finally, the rats were euthanized and brain tissue was collected for biochemical testing or histological analysis. As you can see in the figure 1, experimental design, rats were submitted to a stereotaxic surgery with the injection of AB or saline solution. SAM surgery in the hippocampus, 10 days after surgery, 10 days after surgery. The time required for surgery recovery and AB aggregation. The training period started being physical exercise, PE, cognitive exercise, and none. During 4 weeks, afterward, behavioral tests were performed to evaluate the object and the social recognition memory and to monitor the general behavior. After the euthanasia was performed to brain tissue collection for histological and biochemical analysis. Or object recognition memory test, SR, social recognition memory test, open field plus match. Now the treatments, physical training. Before PE training, the rats were habituated to a treadmill built for rodents to avoid stress effects. The habituation was conducted for 3 days. On the first day, the animals were placed on the treadmill, turned off for 10 minutes. On the second and third days, they were put in treadmill at a velocity of 2 to 5 minutes for 10 minutes. After the rats were subjected to the good runner protocol, which consists of placing the animals on treadmill without inclination for 3 consecutive days, velocity 8 meters per minute for 10 minutes. The level of trainability was evaluated with a score ranging from 1 to 5 points. 1 refuses to run, 2 below the average of runners, runs and stop for runs in wrong direction. Number 3, average runner, 4 the average, runs well with sporadic stops. Good runner, runs and always stays in front part of the treadmill. In the end, the animals that maintained an average of 3 or more points were included in the exercise group. On the last day, VO2, indirect oxygen consumption, running test was performed to determine the individual intensity of exercise. For this, rats started running at a low velocity, which was increased 5 minutes every 3 minutes until the rat was unable to keep running. The time to fatigue and walk volume were considered indirect measures of maximal indirect. The PE started in the following week and was performed at an intensity of 60 to 70% of maximal indirect VO2 3 times a week, once a day for 30 minutes, for 4 weeks without treadmill inclination, physical exercise. Now the cognitive exercise, CE. The CE was based on adaptation of the Barnes maze memory task as proposed. The Barnes maze is a circular platform with 20 potential escape holes equally spaced in the periphery, only one of which leads to an escape cage. Negative reinforcement, bright lights, is used to motivate the animal to escape to a dark cage hidden underneath one of the holes. Visual cues surrounding the maze are used to make spatial learning possible. To perform the CE, the animals were trained every day in the Barnes maze for 4 weeks, and each day they were able to perform the CE more efficiently. That is, they found the escape hole more quickly using the spatial cues. Therefore, every 10 days, the escape cage was altered to another place. Consequently, the animals had to form a new spatial memory, which required cognitive flexibility. Control behavioral task. The open field of elevated plus maze were used to analyze exploratory local motor activities and to evaluate anxiety state respectively. In the OF, the rats were placed in left quadrant of 50 into 50 into 39 centimeter open field made with wood painted white with a frontal glass wall. Black lines were drawn on the floor to divide it into 12 equal quadrants. Crossing and rearing as measures of locomotor and exploratory activities, respectively, were measured over 5 minutes. In the PM, the rats were placed in center of the maze. The maze consists of 2 open arms, 50 into 10 centimeter, and 2 enclosed arms, 50 into 10 into 40 centimeter, with an open roof. Arranged such that the 2 open arms were opposite to each other, the maze is elevated to a height of 50 centimeter. The total number of entries and the time spent in 4 arms were recorded over a 5-minute session. Now the memory test. Object recognition. Rats were first habituated individually to the OR memory task apparatus and left to freely explore it for 20 minutes during 4 consecutive days before the training session. On the 5th day, OR memory training was performed. In the training, 2 different novel objects were placed in the apparatus, and rats were allowed to freely explore them for 5 minutes. 3 hours and 24 hours later, STM and long-term memory, LTM, were evaluated respectively. In its testing session, one of the objects was randomly replaced by a novel unfamiliar object, and the rats were reintroduced into the apparatus for an additional 5-minute period of free exploration. The time spent exploring the familiar and novel objects are recorded. Additionally, the discrimination index, DI, STM and LTM test were determined by the difference of time spent exploring the new T model and the familiar T familiar objects, DI, T novel into T familiar, T novel plus T familiar into 100% and used as memory parameter. Social recognition. SR memory task is an adaptation of social interaction test proposed. The task was completed in 3 days. First, the rats were placed in an arena with 2 small cages for 20 minutes for habituation to the apparatus. On the following day, a training session was performed with the inclusion of one unfamiliar rat in one of the cages for 10 minutes of play exploration. After 24 hours, a testing session was performed when the same rat from the training, now a familiar rat, and a new unfamiliar rat was placed for exploration for 10 minutes. The time spent exploring the familiar and novel rats were recorded. Exploration of the conspecific animal was defined as or touching the small cages with the nose and a forepaws. Additionally, the DI was determined by the difference of time spent exploring the unfamiliar and used as memory parameter. Histological analysis. Some rats, n equals to 4, were anesthetized and transcardially perfused with phosphate buffered saline (PBS) solution followed by 4% formaldehyde. Brains were removed, post-fixed for 24 hours in 4% formaldehyde, and cryopreserved in 30% sucrose overnight at 4 degrees Celsius. Then the brains were frozen, and the coronal brain sections, 12 ohm thickness, were cut in a cryostat Leica CM305OS. The sections were stained by hematoxylin eosin He, and a qualitative analysis of the morphological parameters were observed under optical microscope Olympus CX21. Statistical analysis. First, the data normality was evaluated by the Sapir-Whorf test. Behavioral results are expressed as the mean positive negative, the SD, object exploration time in OR memory, task and rat exploration time in SR memory task were converted into a percentage of total exploration time, and a one sample t-test was used to compare the percentage of the total time of exploration spent on each object rat with a theoretical mean of 50%. The OR-STM-DI and the SR-DI data were compared between the groups using one-way ANOVA. The OR-LTM-DI data were compared between the groups using one-way Kruskal-Wallis followed by the Mann-Whitney test. The OF and PM data were analyzed by one-way ANOVA. The biochemical results that followed a normal distribution, DCFH and TBARS, were compared using ANOVA followed by Tshetiz post HOC test and are expressed as the mean positive negative SD. For non-normal variables, ACHE and FRAP, a Kruskal-Wallis test was performed, followed by Dunn's post-HOC test. These data are expressed as medium positive negative, the interquartile range. The significant label was set at 0.05 for all variables. Now you can see the results in the table below. Control behavioral task. There were no difference between the groups in number of rehearings and the crossings. The figure was 5 by 5.64 equals to 1.171. P was 0.33, and the crossings F5 by 64, 3.279. P was 0.36 during the free exploration session in the OF, showing that treatments and surgery did not affect rats, exploratory and locomotor behavior. In the same way, the procedures did not affect anxiety behavior, since no difference were observed among groups in the PM test F5 by 64, 0.7539 equals to 0.58. These data are important since they guarantee that the results observed in memory tasks are related to procedures influencing learning and memory process and not to other behavioral alterations. As you can see in the table 1, these are the different training and surgery procedures that do not alter the locomotor and expiratory of activities evaluated in the open field and the anxiety behavior evaluated in elevated plus range. You can see the rearings, control, AB, PE, AB plus PE, C, AB plus C, and P value. P value finally goes to 0.33. And the crossings, it goes to 0.09. And the time in open arms, 0.58. Let's quickly move to memory task. These are the graphical representation. Object recognition. In the OR memory training session, the rats explored each object for a similar percentage of total exploration time. In the graph, the data are presented as the mean of all groups. This result was expected since both objects were new to the animals. So the figure represents the hippocampal AB infusion that promotes object recognition, OR memory deficits. PES and CES can revert the OR memory deficit caused by hippocampal AB infusion. The graph from A, it shows OR short-term memory, that is STM. And the B graph shows OR long-term memory, LTM. P, that is 0.05, one sample student test, theoretical mean 50%. Data are presented as mean positive SD, n equals to 11 minus 12 group, TR, that is 20. The white ones are the familiar object, and the black one, which represents the novel object. The same as goes for here. Now in the STM test, the control animals spent more time exploring the novel object, showing that they remembered familiar objects. The same was observed in the PE and CE groups, which spent more time exploring the novel object. The AB infusion impaired STM since the rats spend a similar percentage of time exploring both objects, familiar and novel. The 4 weeks of treatment reversed the deficits caused by AB neurotoxicity. The animals subjected to AB infusion and to PE or CE training were able to form or STM. That is, they explored the novel object more than 50% of the total exploration time. 24 hours after the training, rats in the control PE and CE groups explored the novel object for more than 50% of the total exploration time. The animals in AB group presented impaired LTM since they spent approximately 50% of total exploration time on each object. PE and CE were able to reverse the damage induced by the AB protein since the AB plus PE and AB plus CE groups spent more than 50% of the total exploration time exploring the novel object. Considering the DI used for comparison between the groups, no differences were found in STM test. In contrast, significant differences were found in LTM test. As I showed before you in the graph, the animals of AB group presented lower DI compared to control group. The interventions overcome memory deficits induced by AB protein. No differences were found between control and PE group. Let's move to social recognition, SR. You can see in the graph, the white one is familiar animal and the black one is novel animal. In the SR Memoji test session, control PE and CE rats explored the novel rat for a longer percentage of time than the familiar rat. Animals in the AB group, however, explored each rat for 50% of the total exploration time. PE and CE reversed the deleterious effect of AB protein on SR memory, which is deleted. The memory is deleted. Since the treated animals spent more than 50% of the total exploration time exploring the new rat. So the figure deficit, so the figure like the graph, which represents the effect of hippocampal AB infusion, physical and CE on social recognition, SR memory. The A graph represents the hippocampal AB infusion that promotes SR memory deficit. PE and CE can promote SR memory consolidation in rats that receive intrahippocampal AB infusion. And the B graph, there are no differences on SRDI between the groups. P, that is 0.05, one sample student test, theoretical mean 50%. Data are presented as mean, positive, negative, SD, and 11 months 12 group. Considering the DI used for comparison between the groups, no differences were found. Now the histological results. Morphological differences in hippocampal tissue were observed among the groups. The control group presented a normal structure with a standard organization. PE and CE training groups showed a normal structure similar to the control group. The infusion of AB peptide promoted the formation of vacuole-like structures indicated by arrows. I'll show you the figure. Significant tissue disorganization and clear neuronal tissue loss indicated by triangles in the figure. I'll let you know the figure. AB rats treated with physical and cognitive training showed improvement in hippocampal tissue disorganization due to AB infusion. You can see the figure in next slide. The histological analysis. This is the histological analysis. This is the control. This is for physical exercise. This is for cognitive exercise. This is for AB. This is for AB plus PE. This is for AB plus C. The graph represents the histological analysis. You can see. Finally, the discussion. The present result show the physical NCE can promote recognition, SORT and LTM consolidation in animals with memory deficits induced by AB neurotoxicity. Also, physical and CE were able to reverse long-term object recognition memory, decrease hippocampal lipid perioxidation, and restore hippocampal acetyl cholinesterase activity altered by AB neurotoxicity. Still, PE and CE seem to promote a better morphological organization of the hippocampal tissue, which is altered after AB infusion. It is difficult to study Alzheimer's disease in humans since a precise diagnosis of this pathology is not simple or definitive. On the other hand, there are many available experimental models of AD, each one with benefits and limitations. An ideal model needs to mimic the lesions and symptom of disease in a way that is similar to the real situation. However, currently no animal model reproduces all of the characteristics of AD. As the deposition of extracellular AB is one of the main AD features, brain infusion of AB protein is an important model that can contribute to the understanding of key aspects of AD biology, since it mimics both the cognitive and the oxidative imbalance characteristics of AD. It is important to consider that between the main regions where the AB deposits are in AD patient brain are the hippocampus and cortex, structures that are associated with learning and memory function. Previous studies found increased hippocampal and cortical lipid perioxidation and protein oxidation in AD patients. In the same way, in this study, the hippocampal infusion of AB25 to 35 promoted hippocampal oxidative stress and damage, increased ROS levels and lipid perioxidation, and decreased the total antioxidant capacity and acetylcholinesterase activity in the hippocampus, leading to STM and LTM recognition deficits. Also, in a previous study, our group performed the histopathological analysis. I have showed you the figure of the cerebral cortex of rats submitted to the same AB hippocampal infusion model, and we observed that the cerebral cortex of these rats presented intense deposition of amyloid plaques. The neuroprotective influence of PE on dementia is widely studied. Individuals who practice regular physical activity have a 30 to 40% reduced risk of AD development in comparison to physically inactive individuals. Several studies have demonstrated that aerobic PE, such as running or swimming exercise, improve memory in rats with AB-induced AD. Exercise-induced factors, including neurogenesis, synaptic plasticity, and increased cerebral blood flow, seem to promote beneficial effects on the brain. Following this literature, our results showed that the PE was able to promote STM and LTM consolidation in AB rats, reversing oxidative stress disruption and altering ACHE activity. As impressively as PE training, cognitive training also promoted short and long-term learning in AB rats. There is clinically significant evidence to support the effectiveness of CE, but little evidence about the possible mechanisms involved. In general, this type of intervention aims to maintain cognitive brain function as long as possible, reducing disabilities and improving patients' quality of life. However, our results showed a direct effect of CE on cognition through its influence on hippocampal oxidative balance and ACHE activity. A previous experimental study by our group showed that cognitive training was able to prevent the oxidative damage induced by the AB peptide in same animal model. Here we demonstrated the CE can also have an important role after AB deposition. It is important to highlight that both the physical and cognitive training had a more prominent effect of LTM object recognition memory, since for this type of memory, in addition to ability to learn the task, it is noteworthy that the animals that received the AB infusion and underwent the different training had a significantly higher object DI than those that received beta amyloid but did not perform any type of exercise. The same difference between the groups was not observed in STM OR. It is important to consider the STM and LTM consolidation involve different neurobiological mechanisms as the increase of hippocampal protein and gene expression. That is observed in LTM consolidation but is not required for STM. In this sense, the fact that these different types of memory involve some distinct neurobiological processes can justify the different effects observed. Neurodegeneration related to Alzheimer's disease is most pronounced in hippocampal cholinergic neurons, which are directly related to cognitive function. In rat models of AD that present impairments in learning and memory, a loss of and damage to cholinergic neurons are observed. The degeneration of cholinergic neurons in Alzheimer's disease promotes cholinergic hypofunction, which can result in a decrease of choline acetyltransferase and ACHE activity in the hippocampus. Our data showed that AB promoted hippocampal tissue disorganization, vacuole formation, and neuronal loss. In addition to promoting a decrease in ACHE activity, on the other hand, PE and CE were able to reverse the cholinergic hypofunction caused by AB infusion and to restore hippocampal tissue morphology. Many studies address protection and prevention strategies to avoid AD. The most effective type of strategy seems to act before the pathology. However, we also need to think about treatment for patients who already have the disease, and currently the alternatives are few. Since the symptoms of AD start to appear approximately 20 years after the onset of pathophysiological hallmarks. When the patient discovers the disease, the biochemical changes are already advanced. Currently, pharmacological therapies available for AD contribute only to temporary reduction of symptoms, slowing the progression of the disease. Thus, strategies for treating the disease are important. Here we present two interesting non-pharmacological options that are effective by acting on oxidative balance and cholinergic function. In summary, the model of intrahippocampal A infusion used in this study caused hippocampal oxidative stress and damaged decreased antioxidant capacity, altered hippocampal tissue and morphology, and promoted short and long-term deficits in recognition memory that mimic those described or suggest to occur in AD. Surprisingly, both PE and CE induced memory consolidation in animals that received AD infusion, which normally present deficits. The effect of PE and CE is more prominent in long-term object recognition memory. Still, PE and CE reversed the lipid perioxidation and acetylcholinergic activity, alterations induced by hippocampal AD. Therefore, both PE and CE have the potential to be induced in the treatment of AD, since both interventions could be used in humans. As you can see, some photos from clinical research lab. These are the adult male Vista Rats, which are purchased from Veterinary Hospital, Government of Sikkim, India, and were housed at Research Center Behaviour under controlled temperature, 23 positive negative 2 degrees Celsius in a 12-hour light dark cycle with food and water available. These are the Vista Rats, and it's performed on the animal rights. Thank you.